Our next focus is the maximizing the expression of cloned gene. That we have cloned the gene, although it is expressing, but how we can increase its expression. So we can say that sometime it may be easy that gene it is expressing and its expression it is detectable. But in biotechnological applications or at industrial level, the detectable expression it is not sufficient. ये सिर्फ इसका इल्म होना कि हमारा जो क्लोन्ड डीएनए है वो एक्सप्रेस कर रहा है और कोई प्रोटीन बना रहा है, तो वो ये डिटेक्टेबल एक्सप्रेशन दे रहा है, तो ये एक्सप्रेशन सफिशेंट अगर ना हो तो इसका कोई ज़्यादा इंडस्ट्रियल लेवल पे फ़ायदा नहीं है. So one of the biotechnological applications of gene manipulation is that its expression must be maximized. Although the high expression of cloned gene it is not always desirable because it will put extra burden on the host physiology, but if we control the expression of the gene in most appropriate way, then the high expression it is always desirable at biotechnological level or industrial level. Now the question may be that how we can maximize the expression of cloned gene or the target DNA fragment that has been cloned in an appropriate vector into a suitable host organism. किस तरह से हम उस क्लोन डीएनए का एक्सप्रेशन को हाई कर सकते हैं या अप रेगुलेट कर सकते हैं. तो इसके लिए डिफरेंट फैक्टर्स जो है उनको जहन में रखना पड़ेगा और उनको कंट्रोल करना पड़ेगा. फॉर एग्जांपल, what type of promoters we are going to use to express our target DNA, what may be the initiation codon or its surrounding sequences or the codon of choice. Then the stability of messenger RNA, it is also very important. Plasmid copy number, plasmid stability in the host organism, and again most importantly the host physiology, that how it is disrupting the host physiology after transforming. So all these factors must be taken into account while maximizing the expression of target cloned DNA. And first we will focus on the constructing the optimal promoter, because the expression of every gene it is under the control of promoter. So promoter, if you remember, it is a site where RNA polymerase it can bind and can start the initiation of a target DNA so that it will transcribed into messenger RNA, and then finally protein it is expressed. So different type of promoters has been analyzed and used for the expression of different target genes, and maybe the promoters may be more than 260 or 263 or even more have been reported and compiled, and their sequences have been compared. After the comparison of different type of promoters, it has been found that they have some sort of consensus sequence that has been conserved during the course of evolution, and they are commonly called as -35 region or -10 region, and hopefully you may be well aware from your course of molecular biology. We are not going to discuss in detail. For example, -35 region, it may be having the 5' conserved sequence that is called as TTGACA at -35, and at -10 location the conserved sequence it is commonly called as Pribner box, and it is TATAAT. So these are the conserved sequences of the promoter that are present at -35 or -10 locations. And when we analyze different type of promoters, then we will find that most of the promoters that are commonly used, they are not having uh, much more homology with the consensus sequences, like the lac-operon. It is not having the 100% homology with the consensus sequence that I mentioned, that is minus 35 or minus 10 region, like the trip-operon, a trip promoter, like some lambda PL promoter. So they are not having 100% homology. And it has been reported that if the sequence it is more closely related to the consensus sequence, their expression will be high. So promoter will be much more stronger. To know the activity of the promoter, a system can be used that may be based on galactokinase synthesis. So promoter, it can be combined with gene that is promoterless, like galactokinase gene. The promoter can be inserted, and then the expression of galactokinase protein it is observed. So it is if it is having the high expression, it means that the promoter is strong, and it is having the high homology with the consensus sequence. In addition to that, the space or the nucleotides, number of nucleotides that is present between minus 35 or minus 10 region, they are also very important. For example, it has been reported that कि अगर 17 बेस पेयर का फासला होगा minus 35 या minus 10 के दरमियान, तो प्रोमोटर जो है वो ज़्यादा स्ट्रांग होगा. अगर नंबर ऑफ बेसिस दोनों के दरमियान हाई है, तो प्रोमोटर की स्ट्रेंथ जो है वो वीक होगी. Next, the consensus sequences of different type of promoters. As I told you that it is having minus 35 region, minus 10 region, and this is the consensus sequence TTGACA at minus 35 and TATAAT. So it is the consensus sequence. If the more homology of the promoter is there with this consensus sequence, the promoter will be more stronger. And here lac promoter, it is not having 100% homology with the consensus sequence. On the other hand, this trip promoter, it is having more homology. Last two promoters that are TAC1 or TAC2, they are the more stronger because of high similarity with the minus 35 or minus 10 region. So they are the hybrid promoter that are artificially synthesized and the more stronger one. So here it is the comparison of different natural promoters with two hybrid promoters. And again, if the distance is 17 base pair between minus 35 or minus 10 region, then again the promoter may be much more stronger. So if we are going to use strong promoter, then the expression of the target protein, it may be 20 to 40% of the total proteins that are present in the cell. In addition to that, if cell it is expressing at high level, then there is one disadvantage. The disadvantage may be that ये हाई एक्सप्रेशन की वजह से सेल के ऊपर स्ट्रेस पड़ेगा और कुछ ऐसे म्यूटेंट्स डेवलप हो सकते हैं जो कि प्रोटीन को कम एक्सप्रेस करें या बिल्कुल ही ना करें. तो इस पर्पस के लिए ज़रूरी है कि वी मस्ट मैनेज द कंट्रोलेबल एक्सप्रेशन ऑफ द promoter.